Let's see. Do I control this? I guess that's a general question. All right. So I'm now a synthetic chemist. I was trained as a biophysics. That's what I did my degree in, protein design, X-ray crystallography of proteins. But for the last 10 years, I've been a synthetic organic chemist. I had 100 students running my Organic 1 exam 2 days ago. And so I'll show you what we've been working on the last 5 years. The basic idea is to try and make molecules that can do the things that proteins can do. All right, proteins are these wonderful structures that can catalyze chemical reactions, build structures. They are the molecular machines that make life possible, and I really want to make things like that. And originally, I tried to do that by making unnatural protein. I made one of the first unnatural proteins. It's called 4-HB1. The crystal structure is in the PDB. The thing took me 4 years to put together, and it's a molecular boat anchor. It doesn't do anything, and I know how to make a 4-helix bundle, but I didn't know how to make anything interesting like this, like an enzyme. And so I thought, I don't like folding. I'd like to do something else, come up with another way of making big molecules that can position functional groups, but that are much more engineerable than, proteins, which are like Jell-O. So there are other people who figure this thing out. There are other people who have working on this idea, and this is the general area of foldamers, make new building blocks that are easier to, that fold more readily into three-dimensional structures. And they can primarily make helical structures. I had a different idea. I thought, what if I make building blocks, and instead of making them so they couple through single bonds, like DNA, proteins, carbohydrates, let's make them couple through pairs of bonds. And let's make the building blocks cyclic, so that they have, the core is some ring structure, and then we'll attach two groups on each end of it, so that when we connect them to other building blocks, they will connect through pairs of bonds, and you make a rigid ladder molecule. And then you can use this ladder as a scaffolding to present functional groups the way proteins do, but they don't fold, they just have shape because of the sequence and stereochemistry and structure of the rings. So this is what our building blocks look like. They are bis amino acids, what we call them. They have a cyclic core. They have an amino acid on one side, another amino acid on the other side. Every building block has two stereocenters. You have to synthesize them absolutely stereochemically pure, and you have to be able to make gram quantities. So we've solved all these problems. It costs less than $20 a gram to make this building block. That's starting materials, solvents, silica, everything. And there's only one column involved in the synthesis. And we have lots of these. We have 5-membered rings, 5-6 fused rings, 6-membered rings, different regioisomers. We can make every stereoisomer, 4 of them, of this building block, every stereoisomer of this, 2 of this. We can turn them backwards. We have an enormous amount of backbone control. What we figured out how to do in the last 2 years is how to put functional groups on these things, like amino acids have side chains, and then get the coupling chemistry to work, because that was the problem. Putting functional groups is not the problem. Getting the coupling chemistry to work is the problem, because everything shuts down. All the reactions shut down. We invented a new reaction that solves this problem in a beautiful, elegant way. So now we can make molecules like this that have a rigid backbone, and they present a functional group on every building block. We can control the distance and the relative orientation of these building blocks by controlling the sequence of the monomers, the structures of the rings, and the stereochemistry at all these positions. It really is like a molecular Lego set. Here are the parts, and we can stitch them together in a couple of hours and days to make molecules of almost infinite variety. The way we control shape without folding is like this. This is a chain of bis amino acids. It's a ladder molecule, a spirolateral oligomer. And we can go onto one of these monomers and put up all these black lines. These represent all the different directions the chain could turn if you put one of the other building blocks that we have in this position. So you put another building block in, you click this line, it puts it in. Now you have a hairpin turn. You can go to any other monomer and do the same thing. So we can basically make molecules that follow any path through three-dimensional space you want. It's a single continuous chain. It doesn't fold. It's a ladder, bent, weird, twisted ladder. And we can decorate it with functional groups. We've made more than 100 of these molecules. We're just now starting to develop applications now that we can put a lot of functionality on them, but we've made macrocycles and all sorts of different shapes. We've published about almost 20 papers on this, and we're the only group that's working on it. This is the only thing we do. So one application that we developed was to make molecules that make very large conformational changes, like myosin. So you have a molecule that's got a 2 nanometer rod, a hinge, another 2 nanometer rod, and then put 2 binding groups on the end. And the idea is that when it's unbound, it's floppy and large, and when it binds something, it would close up into a more compact shape. We could use this for all sorts of applications, like sensors. You could put these on nanotubes and put a charge up here. When they bind their analyte, they'll fold over, bring the charge down to the nanotube, and you've got a chemical field effect transistor. We could put these on the surface, and we could detect binding by changes in height with atomic force microscopy. We can put these in nanoscale channels and make valves, active, smart valves that recognize something. If we put boronic acids on here, you add glucose, the glucose binds, folds the thing up. Now you've got a channel that insulin could flow through. You could make a material that acts like an artificial pancreas. So this is the molecule that we made. This is Laura. She's a master's student in my lab a couple of years ago, and she made this molecule, very easy to synthesize. And we put metal binding groups, so it binds copper and folds up. You get very nice binding transition as you add copper. And we use biophysical techniques to characterize these. So when it's open, you use sedimentation velocity experiments, and it falls very slowly through the liquid when you centrifuge it. You add the metal, it closes up and it falls faster. It's like a terminal velocity. With a parachute, you fall slowly. With a parachute closed, you fall very quickly. And we can actually use these numbers to figure out what the axial ratio, the size of the molecule and shape is. And you can see that it goes from a long axial ratio to a compact molecule. Also, if you put it in a size exclusion column, when it's open, it comes out early because it's going around all the little holes in the beads, and when it closes up, it becomes more compact and it gets lost, and it takes longer to get through. So its retention time changes. Now this idea of metal binding, with all these, now that we can put a lot of functional groups on, we can make molecules like this. And I've got a student, a postdoc, who's working on synthesizing this molecule. It displays 3 imidazoles to bind to zinc and leave an open coordination sphere so that it could bind carbon dioxide and perhaps be used to convert carbon dioxide into carbonate, like carbonic anhydrase. And this could be used to capture carbon dioxide for sort of clean coal technology. These things are robust. They do not denature. You could boil them and cool them down, and they're perfectly fine. They're rigid ladder molecules. This is another application we're designing to make a broad spectrum antibiotic by making a triangular molecule that's about 2 nanometers on the side. It's got a hydrophilic core, and these rods are several of our bis amino acids connected together with a hinge on each corner. Put positively charged groups around the outside, and then a gasket of greasy tails. And I think what this thing will do is associate with the anionic membranes of bacteria and then dip down into them and create holes where the bacteria could leak their guts out. This is the principle that our own antimicrobial peptides that our bodies are synthesizing all the time use to kill bacteria. But these things could work at 1/1000th or 10,000th the concentration because they don't have to self-assemble. They're pre-assembled synthetically. So they could work at very low concentrations and be cost-effective broad-spectrum antibiotics. We have a grant from the DoD to develop these as artificial enzymes. So in collaboration with Ken Houck at the UCLA, he designs transition state models, computational models of lowering transition state energies of reactions like this ester hydrolysis, and figures out what functional groups you need in order to accelerate the reaction, to lower that activation barrier. So then what we do is look for bispeptides that can hold the functional groups in space, the same way an active site of an enzyme does, but a tiny little scaffold. And then we are currently developing several of these, but we synthesize the molecule and then we throw the substrate at it and we see if it catalyzes the reaction. If it does, it'll change a lot of things. We have grown crystals of these. These are not diffraction quality, sadly, but we are still working on getting diffraction quality crystals of a bispeptide, but we've done lots of NMR structures. So we could take the structural information from these and feed it back into the design and hopefully one day rationally design catalysts. This is one reaction that we're going after. It's an aromatic Claisen rearrangement, and what happens is you develop a negative charge on this oxygen. And so Ken's group has modeled active sites like this, where you have 2 hydrogen bonding groups that stabilize this negative charge. And so we are making molecules, very simple molecules like this, that hold a carboxylic acid and a phenol to simultaneously hydrogen bond to that oxygen, and then test them for their ability to accelerate the reaction.